I'm Dr. Swasim and welcome to my Derm-classes. In this particular lecture, I'm basically intending to help you interpret a research article on how to analyze the statistical part especially. So let's begin the lecture. So when it comes to the statistical results, including myself, we always look for p-value, and if the p-value is less than 0.05, we just consider the results to be significant or the association to be significant. But actually the question is that enough? Is that just enough? Or just because the p-value is less than 0.05, can we consider the association significant enough, or is there something else that we need to look for? So when it comes to the threats to the validity, the question is there some other factors that could have resulted to the association that you're seeing? So the main errors that can occur is basically the chance factor. For example, the association that you've seen now is just because of chance, and no true association really exists. Or it can be because of any systematic errors, which basically includes bias and confounding. For now, let's delve into detail about the chance. So imagine that you toss a coin and you got 5 times head and 5 times tail. That's just normal. Consider that you got 9 times head and 1 time tail. Can it be due to chance? Yes, it can be due to chance. But can it be because of a defect in the coin structure which has resulted in head turning up 9 times? Yes, it can be. So the chance factor is something that we need to avoid to establish an association. So the p-value just indicates whether the probability of that particular result is due to chance. For example, imagine that the fact that you got 9 times head and 1 time tail, and the p-value was less than 0.05, which indicates the probability of you getting head turned up 9 times due to chance is less than 1 in 20, which means the probability that the head turning up due to chance is very, very less, which means it can be due to some structural problem with the coin. So that's what we are interpreting with the p-value. So I have just simplified the p-value. So if you want to read in depth, you can just go and search the articles which explains what a p-value is. But in simpler terms, this is what p-value stands for. And so to conclude, the p-value basically indicates the probability that the result due to chance is very negligible if it's less than 0.05. Does that completely rule out the fact that it can be due to chance? No, still there is a probability that it can be chance, but generally we consider less than 0.05 as an arbitrary point where you actually consider that this can be because of a significant association and not due to a random error or a chance. Now, what can we do as a researcher to reduce this chance of random error? So when you take the sample sizes, if you can take a large sample size, it actually decreases the chance for the random error. Now that we have addressed the random error, now let's delve into the systematic error. So the systematic error basically consists of bias and confounding. So to interpret them, we need to know what bias and confounding is. So let's start with the bias. So talking about the bias, we have two main forms of bias. One is the selection bias and the other one is the information bias. So what is selection bias? So this is a bias that occurs while you're selecting subjects into your study. Consider that you are analyzing the proportion of advanced stage cancers in a population. What if you selected the subjects from a tertiary care center? It's highly likely that the proportion of advanced stage cancer is going to be higher in a hospital population or a tertiary care center population than in a general population, because the more that you have the symptoms and the more advanced treatment that you have, you're likely to show up in a hospital. So that's a selection bias. When it comes to the case control study, anything that influences the selection of cases and controls other than the exposure results in a bias. So for example, if you're considering smoking causing lung cancer, if at all you select the subjects based on any other influence other than smoking, then that is a bias. What is an information bias? So this occurs while you collect the information. For example, if as a researcher or an investigator, you know that a particular population has lung cancer, you're highly likely to ask them the question if they have smoked. So that is something that gives you an insight into an information bias. So anything that happens while you collect the information is called as an information bias. So what does this indicate? Now as a reader, why you need to know this? So you need to look for these biases, and where can you find the influence of these biases in a research article? You need to check the methodology section, where they would have detailedly written about how they've selected the subjects or how they have collected the information. So that is something that you can interpret, and you can think about the different scenarios that might have introduced the bias. Now, coming to what role does it have in a researcher? As a researcher, you need to analyze these bias and design your study accordingly, because the bias cannot be adjusted in your analysis. So if at all you're not avoiding them in the design of the study, you're going to get results which has influence of other factors in it. So that's what you need to look for, and then you need to properly design your study. So one of the methods that you can use is blinding. For example, if the researcher or the investigator doesn't know that who has or doesn't have lung cancer, it's less likely to have an information bias there. So that's one of the examples. Now, what are the other factors? Now, next is confounding. So, what do you mean by a confounder? So, these are the factors that might have caused the result. For example, take the study where we are analyzing the alcohol causing pancreatic cancer. Can it be a possibility that smoking has caused pancreatic cancer? Yes, it can be. So, how do you avoid that? You need to check if the cases or the controls were smoking. And if smoking was unequally distributed among cases and controls, the fact that the case has got cancer, pancreatic cancer, can be because of smoking. So yes, if everyone's more smoking, both the cases and the controls, then it's most likely that the controls or the cases got pancreatic cancer because of alcohol consumption. So that is an important factor that you need to consider. So what are the properties of a confounding factor? So these are independent risk factors which are distributed differentially across the groups, the cases and the controls, and they are not an effective exposure. What do I mean by they are not an effective exposure? For example, if junk food is causing obesity and we're looking at the association of junk food and hypertension, then probably obesity is just a subsequent effect of junk food, and you can't consider obesity as a separate confounding factor. And so for the reader, we need to check for the confounding factors in a study. So you need to in-depth read the article, especially the methodology parts, and you need to think about what can be the confounding factors that can come up in a study. Now, as a researcher, there are a couple of things that you can do. You can design your study such that you have lessened your confounding factors and you can adjust them in your analysis. So some of the methods you can use while you analyze or design, while you design your study is basically matching, restriction, and randomization. So what do you mean by matching? We just need to match all the factors other than the exposure. For example, if you're checking alcohol and pancreatic cancer, the cases and controls can only be different in terms of alcohol consumption and nothing else. So if cases are smoking, controls need to be smoking. If cases are obese, controls need to be obese. It's just alcohol that should be present in cases and not in controls. So that's about matching. And restriction is when you're restricting the population. For example, as I mentioned at the previous study, you can consider just the tertiary care centers or just the general population, or if you're considering age as a confounding factor, you can just take the younger population and just take the older population. Another thing that you can do is randomization. So randomization or randomized control studies, as we've all heard, is one of the best research methodologies that you can use. So randomization just helps to balance out these confounding factors in general. So these are some of the things that you can use in design of your study. And in the analysis, there are certain methods that we use. So one is called as a stratification, where you analyze different stratas, for example, different age groups. And there is another method called as regression modeling, which is beyond the scope of this. So to summarize, you can't just be looking at the p-value to interpret your article, because p-value only stands for the chance factor, which is a random error that occurs. You need to address the systematic errors that occurs. So the factors that you need to look for are bias and confounding. So as a reader and a researcher, there are certain steps that you need to take while you interpret the articles or while you design and analyze your study. So I've just gone superficially through the factors that you need to check while you interpret the articles. If you need any in-depth lectures on any of the topics that we have discussed so far, please let me know. Thank you so much. Hope this has helped you.